you can see here a picture of 4 children that were born for 2 parents between 1910 and 1920 in New York City. And what's amazing about them, that each one of them got to be 102. The little sister died at 102, 107, 109, and Helen lived till 110. And when I met Helen at her apartment when she was 100 years old, she was smoking. And when I asked her, none of your doctors told you to stop smoking? She said, you know, all 4 doctors who told me to stop smoking, they died. Those are unusual people who live longer and actually live much healthier. They also have a compression of morbidity, so they're not sick much at the end of their lives. And they were an interest to me because of you guys, a nice being with you, because you guys have done a lot of research in animal models and discovered pathway for longevity that we now call the hallmarks of aging. But it was important to translate those to humans and to see, are we talking about the same mechanism, and to find the longevity genes in humans that are, and ask, are they similar to the longevity genes in animals? And now we have 2 large studies. One of them have accumulated 750 centenarians that are genetically homogeneous, kind of like what Miller is talking about, 4-cross hybrids. And also a study taking their offspring and control without longevity and looking what are the phenotypical, genotypical differences between them. And the first thing that I want to impress you by using this data is something that we're all asking and waiting, and is coming in Nature Aging very soon. It's a study with Zhengdong Zhang and other of our collaborators, where we have exome sequencing of all our subjects, almost 3,000 subjects. And in this study, we ask, what are the differences between people with exceptional longevity and between control that probably don't have longevity? And in order to do that, we took all the genotypes in an exome sequencing that were less than 1% of the population, and we find out all those rare variants. But what was important that people did before, they looked at one variant at a time, and we're not built of one variant at a time. So we looked at pathways and assigned them to pathways. And in those pathways, we took some other data and basically got to the pathway analysis to see, are those pathways related to what we find in animals? And you see that up on the top pathways were what we were thinking, we're hoping we will get, the mTOR signaling, the insulin IGF signaling, the MAP kinase signaling, are all very much enriched in centenarians compared to control. So it's kind of a assurance that what we said, that animals aging is conserved, is really true. And that's why we're offering service around this study to all of you. So we have 3 cores in the Nathan Shock Center that I'd like to talk with you about, and I'm going to start with this Human Multi-omics Data Core, because what we have around the centenarians and other studies in human, we have genomics, we have proteomics, we have transcriptomics and metabolomics, and we have lots of cells that we can have. And one of the easy thing that you can do, if you have your favorite genes, if you want to see what's happening to 36, we can throw you all the information and find all the mutations that are probably functional or are highly likely to be functional because we have exome sequencing, and you can analyze and you can ask us what's unique about those people who have mutation in 36 or any other gene of interest. What we have in the core is we have resources from two studies. One is the Longevity Gene Project, which are centenarians and their offspring, and one is from Longevity, which is the longitudinal study. So we have annual follow-up for an average of somewhere like 7 years. And we have a lot of collection. We have neurocognitive assessment and physical assessment and medical, and we have serum and whole blood. We have DNA, we have the genotype, we have lymphoblast cell line, we have a proteomic, we have white blood cells, and we have MRIs and coronary CT of our people. So you can look at your gene of interest, and we can help you give, for example, the cells of the people who have a direct genetic alteration, and you can study that. It's all part of our service. Another core that we have is run by Ana Maria Cuervo. By the way, the other one is by Jan Vig and Sophia Milman. Cuervo runs this core that I think needs less introduction, talking about looking at proteostasis, mainly autophagy, in particular doing a human proteostasis, in other words, in human cells. And we also have Evris Gavatiosis, who's an absolutely incredible medical chemist who designed drugs. And we have 3 drugs that are already in biotechs from the one that he has designed. So those are all services that you can get. I'm not going to go into the assays and reagents, but there's consultation and there's drug development help, and you know the quality of work that Ana Maria and her team are giving. And just to connect between those two cores, we looked at activation of macroautophagy in people without longevity. Those are offspring of parents with usual survival compared to centenarians. And you have to know there's no much difference between 70-year-old and 100-year-old, because the 100 years old are then at the end of their lives. So the offsprings are very important for the phenotype. And you see that basically all the offsprings that we tested in their white blood cells have increased activation of macroautophagy. So those are the kind of studies that we can do, and and you can see how we work together across cores. The third core that we have that is of interest for you is called a healthspan core. And Derek Hoffman, who's a great geroscientist, is doing metabolism, frailty, cognition, and function in animals. And I know that all of you are doing some of that. I'll tell you in a second what he's doing special, but the real special part of the core that a lot of you have been using is chronobiosis. And chronobiosis is not easy to do, okay? Chronobiosis takes time, takes effort, and you have to do it well in order to have healthy animals when you do the chronobiosis. And Derek Lev is incredible with it, and I'm not going to expand here more, but if you're interested, please let us know. By the way, the chronobiosis becomes relevant if you have a mechanism, and you can have a transgenic animals where you, let's say, block the mechanism or whatever, activate the mechanism, and see if this is protecting against the effects of aging or youthfulness, depending on the question you have. So the idea of using the chronobiosis is doing that in transgenic animals to see how the environment affects and if you can protect mechanisms like that. But Derek is doing lots of sophisticated things beside the phenotype, which is cognition too, and echo and body composition and things. He's doing clamps, insulin clamps, but also targeting the hypothalamus. So you can look at a hypothalamic mechanism, which some of you have used, and doing insulin clamps. We have metabolic cages, and we do hormonal assays. And so there's a lot that you can specifically ask here according to the question that you have. Certainly, if you have a mechanism that involves insulin or IGF signaling pathway, and you want to do the state-of-the-art studies, we're kind of the best in doing unstressed animal and insulin clamps. To give you an example of what we have available and how we use it, we have a whole metabolomic panel that is taken for 4 groups of animal. The young animals ad libidum or caloric restriction, and old animals ad libidum or caloric restriction. And what we try to find in a very wide metabolomic panel was things like the example I'm giving here of sarcosine. We're looking for something that declines with aging and that is reversed by caloric restriction. And sarcosine is an example of that. And of course, you can think of other patterns that you're interested to see what happened with caloric restriction. One thing is to decline and have it normalized. The other is to increase and have it decreased. But the interesting thing is that we showed the same in humans in collaboration with Luigi Fontana in his Chronis cohort. We see that all the ad libidum subjects have lower sarcosine level, and with caloric restriction, it's replaced, it's back to normal. And also in Barkey, Ames dwarf mice, Ames mice, whether they are young or old, have higher sarcosine level. And this gave a push to the development of biotech that has a protein, that has a drug that Everest has designed in order to increase level of sarcosine. Just to link it back to aging, again, Ana Maria Cuervo Core has done some autophagy studies and showed that with increasing sarcosine, that there's increase in macroautophagy. So you can see how all those cores are helpful to one another. So this is really the message that I have now. This is how our institute looks like. We have the resources that I told you that are resources that are derived, a lot of them are derived from humans. We have materials, we have data, we have the 3 cores that I described, we have administrative core, and we have research development cores like all of you. A lot of you are collaborating with us, and I would like to collaborate with you. I would like to encourage you. And remember that we have this resource that can help and cross-help all of you. Nice being with you, and now I'll take some questions.